Now that we have fully followed fate number 3 of pyruvate, I want to return back to our big picture view, and I want to follow fate number 1, further oxidation. So now we're assuming that there is a terminal electron acceptor for the electron transport chain, that in fact pyruvate can get further oxidized, and that of course that oxidation is needed, that the cell needs more ATP, and that it needs more reducing power. So in this case, pyruvate is going to get transported in a yeast cell into the mitochondrial matrix, where it is going to get oxidized again, so it will be ripped and stripped of some high energy electrons. Now, if we're following the fate of both of these pyruvates, we would have a stoichiometry of 2 in front of every one of these molecules. And as pyruvate is converted to a 2-carbon molecule, acetyl-CoA, there is the loss of a thoroughly worn out, fully used up carbon dioxide. And it turns out that as pyruvate is converted to acetyl-CoA, the 2-carbon acetyl molecule needs to have a taxicab. This taxicab is called coenzyme A. It is a cofactor, it is a coenzyme, and coenzyme A gets attached onto the 2-carbon molecule in this reaction, which actually is quite a complex reaction. We'll just cover the basics. So let's talk then about the details of that further oxidation, and let's follow the journey as acetyl-CoA then goes to the top of the TCA cycle, where that TCA cycle is turning. Oxidation being the middle name of that TCA cycle, and ripping and stripping and making a lot more reducing power. So as soon as we've covered a brief bit on the transition step, this is what we call the transition step right here, then we'll turn our attention to the TCA and we'll spend some time talking about the details of the TCA cycle. So here we are, our 3-carbon pyruvate getting further oxidized, becoming a 2-carbon acetyl-CoA molecule. And with that, of course, the coenzyme itself has to come in and be added on to the 2-carbon group. There's a loss of 1 carbon, so one of our fully worn out, totally used up CO2 is lost. And of course, reducing power is generated here. So some more NADH that can take its electrons to the ETC and can exchange them at that energy currency exchanger for ATP. This reaction is going to occur in the mitochondrial matrix of cells like our yeast cell, our eukaryotic cell. But of course, in prokaryotic cells, there is no mitochondrion, so this will occur in the cytosol of a prokaryotic cell. Let's follow now the journey of that acetyl-CoA into the TCA cycle. So the TCA, or you may have heard it called by one of its other names, it has many names, the citric acid cycle or the Krebs cycle. This cycle occurs in the mitochondrial matrix of eukaryotic cells, and we saw that, and you'll want to represent that in your poster as you continue to develop it. It actually is a fairly ubiquitous pathway, though it's not entirely ubiquitous. There certainly are some types of microorganisms that lack certain of the enzymes of a fully functional TCA cycle and find other ways to further oxidize their molecules. So there's some diversity in the world of oxidation there. This is what we consider, just like glycolysis, an amphibolic pathway. Because as you might imagine, although the name of the game in TCA is catabolism, breaking down that acetyl-CoA molecule, that is the name of the game. But remember all of the intermediates that take place along the way. And many of those can be used to build other molecules in anabolism or biosynthesis. So we know that many of the intermediates in the TCA cycle can be used as precursor metabolites. And that's why we say that this is an amphibolic pathway, a pathway with a dual nature because of that. So the goal of this pathway, as I said at the heart, is oxidation, stripping and ripping high energy electrons off of the acetyl 2-carbon acetyl group. So complete oxidation of that 2-carbon molecule. So when we see that, and we know that we're going to get the complete oxidation of a 2-carbon molecule, it becomes pretty easy to answer this question. That is, how many thoroughly worn out, totally used up CO2 molecules will be lost in one turn of the cycle? Well, we're fully oxidizing a 2-carbon molecule, so we're going to need to lose 2 carbons. So 2 CO2s will be given off in one turn of the cycle, 2 carbon dioxides. So get ready for that and watch as we go through the turns of the TCA for the production of those 2 CO2 molecules. The first step of the TCA cycle is by far the anomaly. In the first step of the TCA, we see a 4-carbon molecule called oxaloacetate. You can notice there that it has 4 carbons. It's actually going to condense with the 2-carbon acetyl group. This is the only step like this in the TCA. It's the only step where bonds are formed instead of being broken. It's the only condensation reaction. It's the only reaction in which we form a larger molecule. We form a 6-carbon molecule as the 2-carbon molecule and the 4-carbon molecule condense. So the acetyl group is transferred onto the oxaloacetate. This makes a 6-carbon molecule called citrate. And in fact, citrate, the citric acid cycle, right? This is where one of the names of the TCA cycle comes from. So now that we've made this 6-carbon molecule, the second step of the TCA is going to be similar to the second step of glycolysis. Remember how that step was a rearrangement? And there turned out it kind of looked like a boring reaction, but it turned out it's not so boring at all. And that's the truth here as well. Isonitase, the second enzyme, catalyzes the formation of Iso Citrate. This is simply a rearrangement. One 6-carbon molecule gets turned into another 6-carbon molecule. And look, the only difference is that we go from having the alcohol attached to the 3rd carbon to having it attached, the hydroxyl attached to the 2nd carbon. So this goes from being a tertiary alcohol to being a secondary alcohol. And those of you who don't speak chemistry are like, so what, Rachel? Does it really matter to me? Totally understand. But in fact, it does matter because it turns out that a secondary alcohol can be oxidized, whereas a tertiary alcohol is very hard to oxidize. And remember, the middle name of the TCA cycle is oxidation, production of reducing power. So the whole goal is to make a molecule, an intermediate, that can be oxidized. And in fact, that's what step number 3 is all about, oxidation. So isocitrate gets oxidized, and you guessed it, that means that our first NADH pay dirt sort of yield is happening in step number 3 of the TCA. So as isocitrate is oxidized, it forms a molecule called alpha-ketoglutarate. That oxidation yields NADH. So we see the production of our first reducing power. Not our last, because we know a lot of it gets made in the TCA. But notice also that alpha-ketoglutarate is a 5-carbon molecule. So in this process, we also pop off our first thoroughly worn out, totally used up CO2. So if you want the sexy term for what happens in this reaction, it's called an oxidative decarboxylation. You know, next time you don't have something to say when you're out on a date, just belt that one out and your date will be so impressed. Oxidative decarboxylation, right? The formation of reducing power, the concomitant, that means at the same time as production of CO2. So that CO2 gets popped off, oxidative decarboxylation. Now, anytime you see an oxidation reaction, the name of the enzyme is always easy to know. And in fact, that's true of enzymes. You don't need to memorize the name of enzymes. All you need to do is know what they do. And if you know what they do, you can recognize what their names are. So anytime we see an oxidation like this, the name of the enzyme is dehydrogenase. That is the last name. So dehydrogenase is the last name of the enzyme. But then the substrate is the first name of the enzyme. And here, see, the substrate is isocitrate. So we can predict, even before bringing up the name of this enzyme, that the name is isocitrate dehydrogenase. So let's write that down. Now guess what? Name of the game, oxidation. So I bet you guessed number 4, step number 4, another oxidation and another generation of reducing power. So in step number 4, alpha-ketoglutarate gets oxidized, forming the molecule called succinyl-CoA. Now, that means that something else has to happen in this reaction because look who's showing his head again, the taxicab coenzyme A. So just like the transition step, in fact, look at this step. Step number 4 of the TCA cycle is almost exactly the same as the transition step. So a CoA gets added on, and we notice an oxidative decarboxylation taking place again. So we get the production of another NADH molecule, we get the popping off of another thoroughly worn out, totally used up CO2. Hey, check that out. We've now made both of the CO2s that we know are going to come off in the TCA cycle so soon, and yet both of our CO2s have already been made. And I bet all of you can aptly name this enzyme. Name of the substrate, alpha-ketoglutarate, plus last name, dehydrogenase. I'm getting the James Bond thing going through my head, you know, name is dehydrogenase, that is alpha-ketoglutarate dehydrogenase, right? Last name, always dehydrogenase. So now compare this reaction to the transition step. Recognize that it's almost exactly the same. And now go back to the transition step on your previous slide. You can even pause the podcast if you want to. Go back and give a name to the enzyme that catalyzes the transition step. Let me know what that enzyme name is. So now the product of step number 4 of the TCA, it's succinyl-CoA. Some of you might be thinking, man, have we met? Because you really look familiar to me, succinyl-CoA. And that would be because you're doing a good job of recognizing that this is one of the molecules on the phosphoryl group transfer potential table. If you go back to that table, you're going to notice that the overall delta G of hydrolysis of this is actually equal to -32 kilojoule per mole. Wow, it's a whopper, right? Lots of energy in that molecule. And in fact, that's enough energy to make ATP, just barely, but it's enough. So that's what happens in step number 5 of the TCA. It's famous because it's the only step in which ATP is made via substrate level phosphorylation. So as succinyl-CoA is converted to succinate, the energy is harnessed to make ATP. So some of you are thinking, man, you're crazy, Rachel, crazy, because you just said ATP, and I'm clearly seeing a GTP. That's not ATP. But energetically, does it matter? Remember, the energy is in the phosphate groups. So no, it doesn't matter, right? It totally doesn't matter. We can recognize that that has equivalent energy to ATP, and in fact, it can quickly transfer a phosphoryl group onto ADP to make ATP. So pretty easy, quick, smooth transfer, and we get the production of ATP. So this is the energy yielding step, if you will, of the TCA cycle, but that's only because it's the only step in which substrate level phosphorylation takes place. We know that all this reducing power, all this NADH is going to also be worth a lot of energy, assuming that ETC keeps on running, right? The electron transport chain. Okay, so let's go on to the next step of the TCA cycle in which succinate gets oxidized. You guessed it, another oxidation. So I bet we can already name the enzyme that catalyzes this. First name succinate, last name dehydrogenase. So succinate dehydrogenase. Oh, but look, it's an oxidation, so more reducing power is going to get formed. But this time, it's not NADH. Instead, the reducing power that's generated is FADH2. Remember that FADH2, unlike NADH, is not the pizza delivery guy, right? It has to stay attached to its enzyme. So this enzyme, succinate dehydrogenase, I'll just abbreviate dehydrogenase, the FAD is inside of that enzyme and it never leaves that enzyme. So then it has to transfer its electrons over to a Q molecule in order to get them to be mobile. So QH2 is the next mobile carrier of electrons. FADH2 has to stay in its enzyme. Now, ever so conveniently, and some of you know that I like to call succinate dehydrogenase the Wonder Woman enzyme, because as it turns out, succinate dehydrogenase has multiple personalities. That is, by day, it is what is it? Diana, right, is the name of the woman that is Wonder Woman by night. Well, two identities. So we recognize that succinate dehydrogenase catalyzes step number 6 of the TCA cycle. But at the same time, in its moonlighting, it is also complex 2 of the electron transport chain. So this enzyme is both the enzyme of the TCA and an enzyme of the ETC. So that's how in this step number 6, electrons are directly transferred into the electron transport chain in the form of FADH2. Pretty sweet, absolutely my favorite enzyme ever, succinate dehydrogenase. Okay, let's go on to step number 7, where fumarate is hydrated. This forms malate. This seems like a simple step, but in fact it's making the potential for one more oxidation, because malate can be oxidized again, generating one last NADH. I bet some of you have already started to do the math. How many NADHs are made per turn of the TCA cycle? Okay. 1, 2, 3. Wow, a whopper, 3 NADHs per one turn of the TCA cycle. But how many times does the cycle turn for each glucose molecule? It turns 2 times, right? So for glucose molecules, 6 NADHs are generated. That's a lot of reducing power. Also note, don't forget the FADH2. So per turn of the cycle, 1 FADH2 is made. Turns twice per glucose, so 2 FADH2s per glucose. Don't also forget the substrate level GTP. 1 turn, 1 GTP. 2 turns, 2 GTPs. Lot of energy being yielded from this cycle. Let's go ahead and write down that net yield. Oh, and you guys already wrote the enzyme, didn't you? Because you knew that that would be malate dehydrogenase, because malate is the getting oxidized. So last name dehydrogenase, first name malate. Writing down our yield then, we know per turn of the TCA cycle there's 1 ATP, but if it turns twice for a glucose, there are 2. And that, of course, is what happens in step 5. The reducing power, well, we know there's 3 NADHs per turn, or if it turns twice per glucose, there's 6. Holy cow, lots of NADHs. Now, FADH2s, 1 per turn, or 2 per glucose. Wow, a lot of yield, not to mention all the precursor metabolites that can be made in the TCA cycle. For example, alpha-ketoglutarate can get utilized to make glutamate and sparatate. Succinyl-CoA can be utilized to make porphyrins. Oxaloacetate can be utilized to make glucose. So there are a lot of ways in which these intermediates can get siphoned off and be used in anabolism. Think this through. If a lot of intermediates are siphoned away from the TCA cycle, right? If they're siphoned away from that cycle, what does that do to the overall net yield of reducing power and ATP? If you have an answer for that, go ahead and email me. I'll add a point of extra credit for you. Let's do a quick sample calculation as we end off our day. If 1.2 times 10 to the 18th molecules of glucose are catabolized, how many ATP molecules are net via substrate level? Okay, note that, substrate level phosphorylation during both glycolysis and the TCA. Well, this is pretty easy to work out because remember that the net yield of ATP via substrate level phosphorylation in glycolysis is 2 ATPs. Well, remember in the TCA cycle, it's 1 in step 5, but per glucose, it's 2. So essentially, there are 4 ATP made in glycolysis in the TCA via substrate level phosphorylation, and that's all we're caring about right now. So we have 1.2 times 10 to the 18th glucose molecules times 4 ATP per glucose in glycolysis and the TCA. So work that out, and that's your answer. Have a fabulous day, and thanks for such a great Friday lecture. I look forward to seeing all of you on Monday.